Sorry, less time. Let's go. No, actually, what are we doing? We are using ultrasonic fields in liquids to enhance the stability, the sensitivity, and the selectivity of a variety of different inline probes. Yeah. One quick word about the ultrasound. We are using megahertz fields. So these ultrasonic fields are usually safe for sensors, also for cells. And the name is not only a cute wordplay, so the usePAT, it's also an acronym and stands for Ultrasound Enhanced Process Analytical Technology. Now, a quick couple of words about the physics behind it. So if you're using a standing wave field, and a particle enters such an ultrasonic standing wave field, it gets subjected to radiation forces, and those radiation forces force the particles into certain regions. And particles for us is everything that has a boundary to the liquid. So it can be cells, it can be crystals, it can be oil droplets, it can even be air bubbles. Now, we are using this in different applications, but one that I would like to present is this one, where we use this in combination with attenuated total reflection mid-infrared probe. So if you know this technique, you know that you have this ATR crystal here, and the evanescent field only reaches into the liquid for 1 or 2 microns, and you can measure the liquid very well with it. Now, if you couple this with the add-on that we provide with an ultrasonic transducer here, you can create this standing wave field, this ultrasonic trap. You can, for example, catch your cells, and then as you wish, you can push them into the evanescent field or keep them out of it to either measure the cells or measure the liquid. And we did a short application study with this on cyanobacteria fermentation. This was done with the IBD group here in Vienna, the Technical University, with the group of Oliver Spadiut. And what he had here is just a cyanobacteria fermentation where PHP was accumulated inside the cell. And the idea was, can we use this technology to actually measure the PHP content inside the cell, inside the bioreactor? You can see here on the left-hand side, you can see the installation, and wait, so, and you can see our add-on, you can see the probe going in, and you can see the typical glass bioreactor. So what happens if we now iteratively push the cells into the evanescent field, measure them, and keep them away? You can see, for example, here the Mi2 band, it will just fluctuate. So if you push them in, the intensity gets higher. If you push them out, it gets lower. Now, if you make a little trick and just subtract the spectra where we push the cells in from the last spectra where we measured only liquid, we can get actually a clear a pure spectrum, or more or less pure spectrum of the cells themselves. So we can actually see here this distinct band of the PHP. We can see the amide 1 and 2 bands, and we can see the carbohydrates here in the fingerprint region. And if you look at the spectra over the process time, so it's a long process, we can actually see that the PHP is accumulating, or the PHP band is increasing, indicating that the cells actually accumulate PHP, which we also know from the reference measurement. And since it's of limited time, just to show you, this is also available as a free publication. We can actually really monitor this while the process is running and not waiting for the offline samples. So this is a great use of our ultrasonic add-ons to enhance a classical spectroscopic measurement. The second, and commercially for us more important issue, is improving robustness. And by that, we just mean using this technology to clean or keep your probes clean. And the primary application right now for us is a process that might be known to you. It's the activating sludge process in wastewater treatment facilities. And this is a process where you have to do aeration, and you usually use these kinds of DO probes, so dissolved oxygen probes, to measure the DO content in the liquid. And what happens, that when you can see here, this is a quite dirty process, and they just foul over time. That means either you clean very often, or you will have some problems with the measurements. The probe will will underestimate the right content, and you will just put more air into the tank, which is not necessary. And so if you can see here, we put in two of the same probes, and one of the probes is actually our add-on in there. And if you don't use the ultrasonic cleaning, it will just get worse over time. So you will have not the right measurement. Then you can activate the sound and you can actually clean it. It takes a couple of days actually, because it was quite dirty already. But the better use is just to keep it clean. So activate the sound regularly, and you can just have a robust measurement over a very long time. This image is where I always say it says more than 1,000 words, because you can see we actually put on this UV vis probe here, which has here the very narrow slit where actually transmission measurement is taking place. And we have our add-on here, and you can see we get a really clear, or we have a clean probe, whereas the rest of the thing, you can see how in this process the fouling is just covering everything. So yeah, so what I'm researching for, of course, we're always searching for interested end users that have, for example, problems with sensor fouling, but we're also in search for partners for research and development. So if you are like to do some data sets or some projects with us, we can generate data sets for applications. That's always great. And of course, we are also always searching for funded R&D projects. So yeah, I went a little bit too long, but let's see if we have some questions. Thank you.